Hello and welcome back to day 2 of photosynthesis. So we are going to pick up right where we left off. So in day 1 of photosynthesis, we talked about a general overview of what the process is, and then we talked about chloroplasts, and we answered 2 questions. So the 2 questions that we focused on were, why are plants green and why do leaves change colors in the fall? So we talked about the different pigments that plants have. Specifically, we talked about chlorophyll. And so today what we're going to do is we're going to take everything from day 1 and we're going to synthesize it and put it together and actually get into the 2 stages of photosynthesis. So we are going to talk about the light-dependent reactions and the carbon fixation reactions. And again, this is part of an overarching question that we are working on answering, and that question is, how is energy from the sun captured and how is that used in Earth's biosphere? So we are going to start with stage 1. So we said that stage 1 is the light-dependent reactions, and this stage happens in the thylakoids. So we talked a little bit about this in day 1. And this is the photo part of photosynthesis. And so we said that in this stage, light energy is converted to chemical energy. So what we are going to do is we're going to write out the net equation, and then we're going to practice figuring out which parts in the net equation are reduced and which parts are oxidized. So the overall net equation for the light reactions is 12 water plus 12 NADP plus 18 ADP plus 18P, and we are going to use light. So light is our energy source, and then of course chlorophyll will play a role, and we'll talk about the role that that plays in just a little bit. And our products are 6O2 and 12 NADPH and 18 ATP. So in this reaction, if we take a look at our NADP plus goes to NADPH. So if I write up our little cheat sheet here, NADP plus is gaining a hydrogen, which means it is gaining an electron, and so this is a reduction. So we will see NADP plus get reduced. And then on the flip side here, we have water and then we have oxygen. So water is losing hydrogens, so it's losing electrons, and so this is an oxidation. So water is oxidized to oxygen and NADP plus is reduced to NADPH. Okay, so we said that this happens in the thylakoids, and before we actually get into this process and we draw out how the light-dependent reactions occur, we actually have to talk about photosystems. And we're going to talk about the proteins that make up the photosystems and chlorophyll that's in them. That's actually where the light-dependent reactions happen. So let's go ahead and start on a new page here. And it turns out that in plants, there are 2 photosystems. Conveniently for us, they are called photosystem 1 and photosystem 2. However, they do have other names. Photosystem 1 is also referred to as P700, and photosystem 2 is referred to as P680. And these numbers refer to the absorption peaks of these photosystems. So we have an image on the upper right of what a photosystem looks like. So I wanted to include the textbook image because, as you all know, I struggle with drawing, but we are going to recreate this image and we're going to draw our own, and we're going to kind of talk about the pieces. So let's go ahead and draw our photosystem. So here is a photosystem, and again, whether it's photosystem 1 or 2, they look very similar. And a photosystem, because I have not yet actually told you, kind of hinted at it, but I haven't actually told you what it is yet. A photosystem is a protein complex plus pigments. So we're not going to spend a lot of time on the protein complex itself and kind of what proteins make it up, but we are going to talk a little bit about the pigments and what actually happens when those photons reach the photosystem. So in the reaction center of our photosystem, let's go ahead and label this. So our reaction center, these little diamonds, this pair of diamonds that I tried to draw, this is a pair of chlorophyll A molecules. And those are found more or less in the center of the photosystem. And then around the reaction center, we have a bunch of other pigments. So these are going to be our chlorophyll pigments. Now, of course, you and I know from day one that there are other pigments. Chlorophyll is the majority pigment, but we know that there are carotenoids and anthocyanins. Just for simplicity, we're going to leave them out of the image, but do know that they do exist as well in the photosystems. So let's talk about what happens with that photon of light. So we're going to draw our sun that is providing our light energy, and we're going to have a photon of energy, and it's going to be absorbed by one of the chlorophyll molecules. When that happens, we talked about an electron getting excited, so an electron jumping up to one of the higher energy levels. This electron is then passed into the reaction center, and that electron from that reaction center is then passed along. So we talked about how when we have a photon or light energy excite an electron, that electron jumps up and one of two things can happen. That electron can return to ground state, or that electron can be passed along to something else. In this case, in photosynthesis, when this electron is passed along, it's passed along into the electron transport chain. And so photosystems are the complexes that kind of begin this stage. So now that we have talked about photosystems, we have introduced our new energy management molecule that we talked about in day one of photosynthesis. Now we are actually ready to draw out the electron transport chain and chemiosmosis. So as we are drawing this, I'm going to encourage you to think about how what we're drawing relates to and is different from aerobic respiration. So let's go ahead and start with a brand new clean page, because this is going to fill up most of this page. And just to orient ourselves, just like we did with aerobic respiration, we're going to draw our little model of a chloroplast. And when we zoom in and draw our image, we are actually drawing that thylakoid membrane. So what I'm going to do first is draw all of the protein complexes, and then I will draw the thylakoid membrane and we'll label all the pieces. So just because it'll take me a little bit of time to draw these protein complexes, because there are, let's see, 1, 2, 3, 4, 5, 6, 7, 8. There are 8 total protein complexes we're going to draw. I'm going to draw them and not subject you to watching me draw them for a minute. And so the next clip that you see, they'll actually already be drawn, and then we'll talk about them. Okay, so now that we have all of our protein complexes drawn, so you'll see that we have a total of 8. I tried to make some of them different colors. We're not going to give every single protein complex a name just because it'll get complicated really quickly. But we're going to name kind of the key players and the ones that I want you all to focus on. So let's start with our green blobs that we have. So our first green blob that we have is photosystem 2, or P680. Our second one that we have is photosystem 1, or P700. And you might be wondering, Why the heck is photosystem 2 first and then we have photosystem 1? The reason that they're numbered backwards and we start with photosystem 2 and then we go to photosystem 1 is because they were not named by their order. They were actually named by when they were discovered. So we discovered photosystem 1 first, and so since it was a photosystem, we said, hey, let's name it photosystem 1. And then when we discovered the second one, we said, well, of course it's a second photosystem, so let's name it 2. And then when we actually learned how this process works, we realized that photosystem 2 is first and then photosystem 1 is second. So that's why the names are a little bit backwards if you're wondering. And before we start naming the other complexes, let's orient ourselves really quick. So the top of our image will be the stroma, and this is our thylakoid membrane. The inside, so this, I'm going to write interior, this is the thylakoid lumen. So hopefully this gives you some orientation as to where we are. And then let's go ahead and continue naming some of these complexes. The other complex that we're going to name is, this is a cytochrome complex. And you'll see why here in a little bit, why that is important. And we are also going to name this pink one over here. This is ferredoxin NADP plus reductase. So you can probably tell by the name, it has an ACE, that it is an enzyme, and just based on the name NADP plus reductase, you can probably guess that that enzyme is going to be reducing NADP plus. And I'm actually going to redraw this enzyme and draw it a little higher in the membrane. So even though my drawing is not to scale, we want to try to be as accurate as we can be to the process. Now, the very last enzyme that we have on the right, hopefully you all recognize that. This is our ATP synthase. So same enzyme that we had in aerobic respiration, and this is our proton channel. All right, so now we have all of the major key players labeled. The last thing that we're going to do in this image is we're going to label the parts that are part of the electron transport chain. So the first half, well, more than half, the first left side of this image is the electron transport chain. So just like we had in aerobic respiration. And then this right half over here is where the chemiosmosis occurs, and that's going to be with our ATP synthase. So just knowing what you know about aerobic respiration, you can probably start to hypothesize what's going to happen. We're going to pass electrons through the electron transport chain. We're going to have movement of protons to create a proton gradient. Those protons, now because we have a proton gradient, we have potential energy, and the flow of those protons down their concentration gradient will power the synthesis of ATP. So we're going to make ATP and we're going to reduce NADH. So the question becomes, well, where is that energy coming from? And we've sort of already answered that. So let's go ahead and draw our sun up here. So we said in this stage, we have an input of energy. So here is our light energy that is made up of photons. When this happens, we talked about how in our photosystems there's that reaction center. So a pigment of chlorophyll will absorb that light energy, and an electron will get excited. That electron will be passed to the reaction center, and then that electron is then passed into the electron transport chain. So our electron is going to get excited, and it's going to get passed into that electron transport chain. So let's go ahead and summarize what we've said so far. So a pigment absorbs a photon, and then an electron is excited and passed into, I'm going to write ETC for electron transport chain. Well, now we kind of have a problem, right? So we've taken this electron and it's been passed into the electron transport chain. So now we have this photosystem, so PS2, and I'm going to write PS2 plus because it's lost an electron. It is so strong of an oxidizer, because again, it lost that electron, that what it does is one of the most amazing processes on this planet is it takes water, and that water is split into 2H plus half of an O2. And again, the reason that this happens is because this photosystem 2 plus, which is oxidized, is then going to split water. And so we call this process of splitting water photolysis. And the name photolysis is kind of misleading, and photolysis is defined as light splitting water. And the reason that this is misleading is because it's not necessarily the light itself that is splitting the water, but the reason we call it photolysis is because the initial energy that passes on that electron, well, that excites that electron that then gets passed on, comes from light, and then because that photosystem, that P680, is oxidized and is such a strong oxidizer, it then splits the water. So that's why we call it photolysis. Again, a little misleading, but that is the, unfortunately, that's the definition that we have. So, all right, now that we have split water, well, the reason that we split that water is we split the water so that we can then take the electrons from water. So you'll notice that when we split water, we get 2H plus half of an O2. So those 2H pluses, those protons, each one of them has lost an electron, and those electrons are then taken by photosystem 2, so that now we can go back to photosystem 2 without any sort of charge. So it's happy because it got its electrons back. So now if we hop back into this complex here, we started shuffling those electrons. They're going to continue to be shuffled along until we get into photosystem 1. Now, as these electrons are passed through the electron transport chain here, you'll notice that we also labeled the cytochrome complex. And the reason we gave the cytochrome complex a name is because it plays a very important role, and that role that it plays is it pumps protons into the lumen. So as we have redox reactions that are occurring, those are exergonic redox reactions. So these exergonic redox reactions, and again, this comes from as the electron is passed through the electron transport chain. So as these occur, these are providing the energy that is needed to pump those protons from the stroma into the lumen. And just like we had in aerobic respiration, where we're pumping those protons, the goal is to create a high proton gradient, or a high proton concentration to create a proton gradient. Okay, so now let's go back to our photosystem 2. So, or I apologize, our photosystem 1. So here in photosystem 1 now, we have this electron that has been passed to it, and this electron now has a much less energy than it did when it initially entered the electron transport chain. So at this step, we get another photon and more light energy, and the same thing happens again. That electron is then excited And because that electron gets excited, you probably guessed it, it continues to then be passed through the electron transport chain. So let's go ahead and jot that down here in our image as well. So we have another pigment that absorbs a photon, and then that electron is excited and passed in the electron transport chain. And at this point, we actually have had 2 electrons that have been passed through the electron transport chain. One of them comes from photosystem 2, and one of them comes from photosystem 1. And photosystem 2, its electron gets replaced here by water when we split it. And it turns out that for photosystem 1, that electron is also replaced from photosystem 2, which comes from that water. So those 2 protons that we have right there. So by the end of this, both photosystem 2 and photosystem 1, even though they've passed, each of them has passed an electron down the electron transport chain, each of them actually gets an electron from that photolysis that happens. So now we have finally made it to the last complex before ATP synthase. So this ferredoxin NADP+ reductase. And this is going to be our final electron acceptor. So this electron has been shuffled through a series of redox reactions through these exergonic processes. And when we get to the ferredoxin NADP+ reductase, NADP+ is then reduced to NADPH. So this is where our NADPH comes from. And now that we have covered the electron transport chain part of this process, now we can talk about the chemiosmosis. So while these electrons have been shuffled through the electron transport chain, our cytochrome complex has been busy pumping protons from the stroma into the lumen. So what we have because of that is we have a high concentration of protons inside of the lumen and a low concentration of protons in the stroma. And you can probably guess, based on what happened in aerobic respiration, what's going to happen in this chemiosmosis process. We are going to have a proton that will enter the proton channel, and it will go through ATP synthase, which will cause ATP synthase to rotate, which will cause ADP plus P to make ATP. And because scientists have to have a special name for absolutely everything, the process of making ATP in this fashion has a special name, and that special name is called photophosphorylation. So in aerobic respiration, we called it oxidative phosphorylation. In photosynthesis, we call it photophosphorylation. And photophosphorylation simply means that the synthesis of ATP is coupled to the transport of electrons that were energized by light. Okay, so we've got a lot going on this page, and before we move on, we're going to walk through this one more time because, again, there are a lot of pieces. So we have a total of 8 complexes in this image, and we're mostly focusing on photosystem 2, the cytochrome complex, photosystem 1, the ferredoxin NADP+ reductase, and ATP synthase. So we're going to start on the left and work our way to the right. And again, I am just re-summarizing and re-saying what we drew just to make sure that we're all on the same page. So starting here with photosystem 2, or P680. Photosystem 2 is made up of many proteins and many pigments. And when we have light energy, and those photons of light, they are absorbed by a pigment in P680. That pigment, an electron in that pigment then gets excited. That electron is passed into the reaction center, which is made up of 2 chlorophyll molecules, and those then pass that electron into the electron transport chain. When this happens, our P680 is oxidized, so it has now lost an electron and it wants to replace the electron that it gave up that went into the electron transport chain. And so we have an enzyme that is then responsible for taking water, and it splits that water into 2 protons and half of an oxygen molecule. And those electrons are then going to go into photosystem 2. One of them replaces the electron that photosystem 2 gave up. The other one gets shuffled through the electron transport chain because it's going to make its way to photosystem 1. So these electrons are now going through these complexes. As these electrons are passed through the electron transport chain, just like in aerobic respiration, we have a series of redox reactions that occur in every single complex. Because these redox reactions are exergonic, they are going to provide the energy that the cytochrome complex needs to then take these protons and to pump them into the thylakoid lumen. So those electrons are shuffled in the electron transport chain, protons are pumped into the lumen, and eventually as we get to photosystem 1, those electrons have now reached a ground state. They're no longer as excited, but we still got some shuffling to do through that electron transport chain. So we have another photon of energy that is absorbed, this time by P700. So again, we have a pigment in that photosystem that absorbs the photon, an electron gets excited, hops up to the higher level, and then gets passed into the electron transport chain again. And so as it's passed into this electron transport chain, again, we have a series of redox reactions. And because photosystem 1, one of those chlorophylls gave up its electron to go into the electron transport chain, so that excited electron, that electron is then replaced from one of those electrons that were originally from the splitting of water. So now both P680 and P700 both have gotten back their electron that they originally gave up into the electron transport chain. So almost done here with the electron transport chain. This electron finally makes it to this last complex, this ferredoxin NADP+ reductase. And that electron is then given to NADP+, and NADP+ is reduced to NADPH. And finally, now we can talk about the chemiosmosis part. So through this entire process, through these redox reactions, we have been busy pumping those protons into the lumen. So now we have this high concentration of protons in the lumen, a low concentration in the stroma. And now that we have this concentration gradient, which is a form of potential energy, these protons, as they diffuse down, so as they go down their concentration gradient, the only way they can get across the thylakoid membrane is through ATP synthase. So they go through this proton channel, they're going to be swung around, and it's going to power the ATP synthase motor. And as that motor is turning, that allows it to make ATP, and we call this process photophosphorylation. So again, looking at this, hopefully you see the similarities and the differences between aerobic and anaerobic respiration. So this process of electrons being passed along is sometimes also known as the Z scheme, and it's essentially another way to look at stage 1. It leaves out the chemiosmosis process, so it's really only looking at the electron transport chain, but it is definitely a much more simplified version. So we're going to draw the Z scheme, and we're going to look at the energy and the redox reactions that occur. So we're going to start with a brand new clean page. So let's go ahead and we are going to draw ourselves a nice X and Y axes here. And apparently today's another day where I'm going to struggle with straight lines. Okay, there we go. Much better. So we have free energy on our y-axis, and we're going to put time on the x-axis. So we're going to start with our photosystems. So let's get those drawn on here. And I'm going to draw our diamonds, which represent our reaction centers. And let's go ahead and get these labeled too. So here is our P680, which is photosystem 2. And our P700, our photosystem 1. So we're going to go ahead and draw our sun that represents where our energy is coming from. We're going to have a photon that is absorbed, and when that happens, we have an electron that is excited that gets passed into the reaction center, and that electron is then given up into the electron transport chain. So sometimes we write this photosystem as P680 with an asterisk, and this asterisk represents an excited photosystem. And so here is our really excited electron. That electron is then passed through a series of redox reactions. So we're kind of leaving off the complexes here. We're only drawing the photosystems, and eventually it's reaching a lower energy state as it reaches P700. While this is happening, we also have water that is being split into protons and oxygen, and 2 electrons are passed into P680, and those 2 electrons will replace the electrons that the photosystems gave up to go into the electron transport chain. So now that we have reached our P700, we have another photon of light, and we kind of repeat this process. So we get an electron that is excited. That electron then gets passed into the electron transport chain. So again, sometimes we refer to this as P700 with an asterisk indicating that it's an excited photosystem, and that photosystem then gets an electron back from that photolysis that occurred. And then we pass that electron until we get to the ferredoxin reductase complex that takes NADP+ and reduces it to NADPH. And so we call this the Z scheme because if you kind of turn your head to the left about 90 degrees, it looks like a Z. The way that it's drawn, it looks more like an N. Where the name Z scheme comes from, I'm not 100% sure. But again, if you turn your head, it kind of looks like a Z, so I suspect that's why it is named that way. So the Z scheme is just kind of talking about this flow of electrons and the free energy associated with that. So this diagram really only focuses on this part of the process. So just the electron transport chain part of stage 1. So it's a little bit cleaner to look at it this way than the previous picture because we have a lot of things going on in our drawing. But they're both saying very similar things. So believe it or not, that wraps up stage 1. So just like we did in aerobic respiration, we're actually going to do a summary at the end of each stage. So for the location, this happens in the thylakoids. And what goes in? We have water that goes in. We have photons, so our light energy. We have ADP and NADP+. And what comes out? We get oxygen, and our total ATP is 18 ATP and 12 NADPH. And if we were to do a one-sentence summary to summarize what happens in stage 1, we could say that energy from sunlight is captured and stored. And when we store it as chemical energy. And that chemical energy is stored in the energy management molecules. All right, so now we are ready for stage 2, which is known as the carbon fixation reactions, and it's also known as the Calving cycle. So the name Calving cycle actually comes from Melvin Calvin. So here on the right, this is a picture of Melvin Calvin, and he actually figured out how the entire Calving cycle works. And he won the Nobel Prize in Chemistry in 1961. So the Calving cycle is actually named after him. And just like we did with stage 1, where we started with a net equation, we're going to start with a net equation for the Calving cycle as well. So we're going to begin with those 12 NADPH and with 18 ATP. So these are the energy management molecules that we made in stage 1. And we're going to add 6 CO2. And then our products are C6H12O6 plus 12 NADP plus 18P plus 18 ADP plus water. So again, this is our overall net equation. And I started kind of running out of room there, which is why I had to go down a little bit. And just like we did with our stage 1, we're going to practice what is oxidized and what is reduced. So I always look for my energy management molecules, specifically my NADH in aerobic respiration, FADH2 in aerobic respiration, and NADPH in photosynthesis. So here's our NADPH and here's our NADP+. So from reactants to products, we lose a hydrogen, which is losing an electron, and so this is oxidized in the process of the Calving cycle. We can also take a look at our CO2 here and our C6H12O6, or our glucose. So our 6CO2, we take that carbon and we fix it to make our carbohydrate. And when we do this, this is reduced. So our 6CO2 are reduced to glucose in the Calving cycle. And again, hopefully as we go through this, I encourage you to think about how this is similar and how this is different to aerobic respiration. So we can actually draw our energy reaction coordinate diagrams to summarize and remind ourselves of what's going on here. So let's go ahead and start with our ATP and our NADPH. So our ATP and our NADPH are used, so those energy management molecules, and we took that light energy and we stored that energy as chemical energy. So now in stage 2, we are then taking that chemical energy and we're going to extract that, and we're going to use it to take CO2, and we're going to steal those carbons, and we're going to fix them to make glucose. So that huge endergonic reaction of making glucose and those carbons coming from CO2 is going to be powered by stage 1. And stage 1 again produce those energy management molecules. So now that we've drawn that, we've gone through our net equation, now we're ready to talk about what actually happens in the Calving cycle. And I like to compare the Calving cycle to the citric acid cycle. So in the citric acid cycle, hopefully you all can quickly summarize it, and that's exactly what I'm going to do here. So in the citric acid cycle, we talked about how we take an acetyl group, and that acetyl group comes from acetyl-CoA, and we combine it with a 4-carbon molecule known as oxaloacetate. And this makes citrate. And then we use that citrate and it goes around in a circle, and essentially what we do is, using enzymes, we knock off a carbon at a time. And that carbon that is knocked off, so we lose 2 CO2s, so lose 2 carbons. Those carbons originally came from glucose. And so at the end of the cycle, we are left with oxaloacetate. So that oxaloacetate leads a really boring life. It kind of just goes around in a circle. It's combined with an acetyl group. It makes citrate. We use a bunch of enzymes. It undergoes structural rearrangements. We lose 2 CO2. And then at the end of the citric acid cycle, we get oxaloacetate again, and we repeat the process. And while we're doing it, so during this time, as we are breaking that acetyl group down further, so again, it came originally from glucose, so we're continuing to break down glucose. So this is an exergonic reaction. We're coupling that exergonic reaction, and we're transferring that energy to energy management molecules. So during the citric acid cycle, we reduce NAD plus to NADH, we reduce FAD to FADH2, and we make ATP. So why am I talking about the citric acid cycle when we're supposed to be talking about the Calving cycle? The reason is because if you understand the citric acid cycle, you can understand the Calving cycle. They're essentially backwards. So instead of transferring energy to energy management molecules, we are taking and using the energy management molecules from stage 1. So that's going to power the Calving cycle. And then instead of releasing CO2, which is coming from the breakdown of glucose, we are going to use the carbon from CO2, and we're going to combine it with a 5-carbon molecule. So in the citric acid cycle, we combined the acetyl group, which is left over from glucose, and combined it with oxaloacetate, which goes around in a circle. In the Calving cycle, we're taking the carbon from the CO2, we're combining it with a 5-carbon molecule that'll go around in a circle, so it leads a very boring lifestyle as well. And the goal here is to make or to build sugars. So that's why I say that the Calving cycle is backwards compared to the citric acid cycle. They both have a molecule that combines with carbons and goes around in a circle, but we're doing things opposite. So in the citric acid cycle, we're harnessing energy from the breakdown of glucose to make energy management molecules. In the Calving cycle, we're using energy management molecules from previous stages to then build sugars. So now we are ready for the Calving cycle. And just like we did with the citric acid cycle, we're actually going to start with a general overview of the Calving cycle. We're going to draw it linearly, and then we're going to talk about counting carbons, and you will see why in a second. So let's do an overall summary. So we're going to draw our carbons just as circles or as balls like we have been. So in the Calving cycle, we start with CO2, and we combine that with a 5-carbon molecule, and that 5-carbon molecule is called RUBP. And RUBP stands for ribulose bisphosphate. So the ribulose bisphosphate, you can think of it as being equivalent to our oxaloacetate. It's what starts the cycle and what that carbon is attached to. So using one of the coolest enzymes on the planet, and it is called Rubisco. Rubisco is able to take that carbon from CO2 and RUBP, and it welds it together to make what we're going to just refer to as a 6-carbon unstable intermediate. And just like in glycolysis, where we have a 6-carbon unstable intermediate, we're going to break that sucker in half. So when we break that in half, we end up with 2 molecules of PGA. PGA stands for phosphoglycerate. And when we have phosphoglycerate, we are actually going to take that phosphoglycerate, and using enzymes, it's going to be converted to 2 molecules of G3P. Now, when we convert this doesn't come easy. This is not a cheap process. This process requires 2 NADPH to be oxidized. So that's where 2 of our NADPHs that we made come from. In addition to NADPH, it also requires the investment of 2 ATP to be used as well. So we use 2 NADPH and 2 ATP, and we make G3P. Now wait a minute. You are probably saying, wait, G3P sounds really dang familiar. And it should. And if you recall back to glycolysis, and when I'm editing this, I'll actually insert our picture that we drew to remind you of this. So when we first talk about glycolysis, we talk about the energy investment stage where we take glucose, we add 2 phosphates onto it, we get this like unstable 6-carbon intermediate, we break it in half, and we get G3P. If you're wondering, is this the same G3P? Yes, it is. So this G3P, we can then go ahead and some G3P can leave the cycle. And when that G3P leaves the cycle, we can take 2 G3P to make 1 glucose. Because remember, glucose is a 6-carbon molecule. And we also take some of that G3P and we have to remake our RUBP. Because if you recall, in the citric acid cycle, oxaloacetate was always remade, and it always went around and around and around in a circle. RUBP does the exact same thing. However, when we make our RUBP, we also have to use a molecule of ATP to remake that. So as you're looking at this, hopefully you've got an alarm going off in your head. And the alarm is probably saying, well, something doesn't quite add up, right? We kind of have a problem here. So our problem is that we need to remake RUBP. But RUBP is a 5-carbon molecule. G3P is a 3-carbon molecule. So if you look at this overall summary that we drew, here we have 5 carbons and here we have 3. That's 8 total. But we started with 1 carbon from CO2 and 5 carbons from RUBP. So something here isn't adding up, right? So you're absolutely correct. And you're probably thinking, okay, well, so how does this happen? The way that this happens is we actually have to go through the Calvin cycle multiple times. So what we're going to do is we're going to start with a blank page, and we're going to talk about this problem, and we're going to work through it. So I'm going to use a different color for every single turn of the Calvin cycle. And then on the right here, we are going to have RUBP that is used and the number that we need to remake. So remember that this is a 5 carbon molecule. So again, this will just be where we keep track of how many RUBPs we have made in each of our turns. So let's start with turn 1 of the cycle. So in turn 1 of the cycle, we are going to start with CO2. We're going to take RUBP. Using that really cool enzyme Rubisco, we make a 6 carbon unstable intermediate. That's going to be split in half, and eventually we make G3Ps. So I'm kind of simplifying the steps here, again, just to illustrate a point. So let's go ahead and talk about the number of total carbons at the end of one turn of the Calvin cycle. We have 6 carbons total. So at the end of one turn of the Calvin cycle, we make 2 G3Ps. Each G3P is made up of 3 carbons. Therefore, we have 6 carbons total. So for one turn of the Calvin cycle, we have used 1 RUBP, and we need to make or remake our RUBP. So at this point, we have no G3P that can leave the cycle. So that's all right. Let's go ahead and let's do this again. So what happens if we do a turn 2? So we're going to go through the Calvin cycle yet again. We're going to take a molecule of CO2, take a molecule of RUBP, make a 6 carbon unstable intermediate, break that in half, eventually make 2 G3Ps. And let's go ahead and add a tally here. So now at this point, if we count the total number of carbons from turn 1 and turn 2, we have 3, 6, 9, 12. We have 12 carbons. So now at this point, we have used two RUBPs, both of which require 5 carbons. And so that means of these 12 total carbons, 10 carbons would have to go to make RUBP, which would leave 2 carbons left over. But G3P is a 3 carbon molecule, so we have no G3P that can leave the cycle. So all right, we're gonna do this again. So let's do turn number 3 of the cycle. And we're gonna add a tally here because we're gonna use another RUBP. So we're gonna take CO2, add RUBP, make our unstable 6 carbon intermediate. That is going to break in half. And now let's go ahead and tally up our total carbons. So from turn 1, we have 6. From turn 2, we have another 6. And turn 3, we have 6. So we have 18 total carbons. So at this point now, if we calculate our carbons, we should have enough to remake our RUBPs and to make 1 G3P molecule. So we used 3 RUBPs multiplied by 5 carbons, which means we need 15 carbons, and these 15 carbons are going to remake 3 RUBPs. And then when we take 18 minus 15, we get 3 carbons left over, and this is going to be 1 molecule of G3P. So if we were to summarize this and talk about and write up a key point. So why did we go through all of this? The key point here is to show you that for every 1 molecule of G3P, we must do the Calvin cycle 3 times. And the reason for that is to regenerate that RUBP. If we were to stop at turn 1 and have those G3Ps leave the cycle, we would never be able to continue doing the Calvin cycle because eventually the cell would run out of RUBP. The Calvin cycle would stop, which means that the plant cell would never be able to make any sort of sugars and eventually would die. And so again, just like we have to recycle oxaloacetate, we have to recycle NAD plus in fermentation. We also have to recycle or RUBP, otherwise the cell will stop working. So on this page, I actually have a video, and I'm gonna write, please watch the video. And unfortunately, I can't show it. The last time I showed a video in our recorded lecture, YouTube yelled at me because it was copyrighted. And so I don't want to get in trouble and have these lectures taken down. But the video is a TED Talk. It's about 5 minutes long. And it does a really nice job of taking what we just talked about and showing it to you in 3D form and in cartoon. And so again, I really encourage you to watch the video. If I could show it, I would, but I don't want YouTube to get angry at me again. So this is one way of looking at the Calvin cycle. And just like we drew a circular version of the citric acid cycle, we're going to do the same for the Calvin cycle. All right, so far in our conversations about the Calvin cycle, we have been talking about the Calvin cycle in a linear fashion. But because it's called a cycle, we know that it is cyclical. So we're going to take all of the information that we just drew on our previous pages, and we're going to summarize it into a simplified model of the Calvin cycle. So there's actually a PDF of this handout, and it is posted on Moodle. So if you would like, when you're taking your notes, you can print it out and you can use that, or if you want to print it out later to practice, it is available there for you. So one of the things I have not yet told you about the Calvin cycle is that it has 3 stages. Stage 1 is called fixation, stage 2 is called reduction, and stage 3 is called regeneration. And as we walk through the Calvin cycle, we'll talk about what that means. So we're going to start on the left with the little rectangular box. This rectangular box is going to represent our light reactions. So this is, of course, stage 1. In stage 1, we form 2 things. So we make NADPH, and the other thing that we make is ATP. So we harness the energy from light and we transform that energy into chemical energy that is stored in the bonds of NADPH and ATP. So to start the Calvin cycle, we need a couple of things. The first thing that we need is we need CO2. And for this model of the Calvin cycle, we're summarizing 3 turns. We just illustrated how we need 3 turns to make 1 G3P, which is why we're going to write 3 times for everything and just summarize it into 3 cranks of that Calvin cycle. So we require 3 CO2. The other thing that we need is we also need RUBP. So let's go ahead and draw our RUBP here. And the enzyme, so I'm going to go ahead and add Rubisco onto here. So Rubisco is going to take RUBP and CO2, and it's going to fix that carbon. So it's going to take that carbon from CO2 and steal it essentially and then knock it onto RUBP. And so that is going to form 3 times of a 6-carbon unstable intermediate. That 6-carbon unstable intermediate is then going to split in half. When it splits in half, we get PGA molecules. So at this point, we talked about how PGA is then taken and PGA is going to be converted into G3P. And if you recall from glycolysis, we've been a little lazy, but those G3P molecules actually have a phosphate group on them. So when that PGA is reduced to G3P, we actually need an input of energy. So recall that a reduction is an endergonic reaction, and so we're going to need some exergonic reactions so that we can couple those not favorable endergonic reactions to a favorable reaction. So let's go ahead and jot down that PGA. Is reduced to G3P. And if you are wondering where that energy comes from, well, right here, right? We just spent all of stage 1 going through the electron transport chain and making a bunch of energy management molecules by using light energy. So we're going to use 2 ATP. Those are going to be hydrolyzed. And that's going to happen 3 times because, again, we're doing 3 turns of the Calving cycle. We are also going to take NADPH, and we're going to use 2 molecules of NADPH, and those are going to be oxidized to NADP+. And these energy management molecules, so those that chemical energy that was stored in those bonds of ATP and NADPH is what powers the reduction of PGA to G3P. So the reason that stage 2 is called reduction is because we are reducing PGA into G3P. So G3P is our golden molecule. It's the one that we are after. And we talked about how for 3 turns of the Calving cycle, we can have 1 molecule of G3P leave the cycle. That molecule of G3P can then be used to make glucose and other carbs. So now what we are left with is 5 G3P molecules, because we had 6 at the end of stage 2, 1 of them left, and now we have 5 G3P molecules, which means we have 15 carbons, the exact amount that we need to regenerate our RUBP. But we are remaking RUBP, which requires a net input of energy. And so with the leftover ATP, we are going to take 1 ATP per turn, and that is going to be hydrolyzed to power taking these G3P molecules and remaking RUBP. Now, if you were to count up the total of NADPHs that are used and the total ATPs, you will calculate that 12 NADPH molecules are used to make a glucose molecule, and 18 ATP are used to make a glucose molecule as well. So if we take a look at our NADPH right here, we have 2 times 3, which is 6, and then we have to do the Calving cycle 6 times total to get 2 G3P molecules to get 1 glucose. So 3 times 2 is 6, times 2 is 12, so there's our 12 NADPH. We have 3 times 2 ATP is 6, and then 3 times 1 is 3. So we have 9 ATP molecules used in 3 turns, but we have to do 3 more turns to get 1 more molecule of G3P to make 1 glucose. So that 9 times 2 is 18 molecules of ATP. So again, I encourage you to print this handout on Moodle and to go through and kind of quiz yourself on this. Alternatively, you can also use this method that we have where we drew the overall summary. I like to present it both ways because I think the linear process shows us the problem that we have, but I think the linear process, but I think that the circular process here, the cyclical process, reinforces that it is in fact a cycle. So now what we're going to do is we're going to answer a really quick practice problem. And I've kind of already given away the answer. And this question is asking us, how many turns of the Calvin cycle do we need to do to get 1 molecule of glucose? So recall that we need 2 turns for 1 molecule of G3P. And we need 2 molecules of G3P to make 1 glucose. So if we need 3 turns for 1 molecule of G3P, that means that we have to do that twice. So 3 times 2 is 6. Therefore, 6 turns of the Calvin cycle will get us 1 molecule of glucose. So now we are ready to summarize stage 2. So stage 2 happens in the stroma of our chloroplast. And we have CO2 that goes in, and our NADPH and ATP that was made in stage 1. And then what comes out is G3P to make glucose. If we were to summarize in general what happens, we can state that carbon from CO2 is fixed and G3P is produced, and this is powered by our energy management molecules from stage 1. All right, so we have a couple more things that we're going to do before we end this lecture. I apologize. It's probably going to be a little longer, but I promise the next topic is going to be a little bit shorter. So let's go ahead and summarize photosynthesis. So I really like this image because it kind of puts all of the pieces together. So it shows us here the thylakoid membrane, it shows us the stroma out here, and the lumen on the inside. So for stage 1, the light-dependent reactions, we transfer energy from light to our energy management molecules. In stage 2, we use the energy from stage 1 and CO2 to make G3P. So the reason that I like this image is because it shows us how stage 1 and stage 2 are connected. So here in the light-dependent reactions, here's our NADPH that was reduced. Here's our ATP that is made through chemiosmosis. And here in the Calvin cycle, or in stage 2, we use this ATP and we use this NADPH, so that energy that is stored in those chemical bonds, that is used along with CO2 to make these organic molecules, which of course is our G3P that can then be used to make glucose. So what we're going to do is we are going to do a couple of practice problems. So the first practice problem that we're going to do is a comparison of aerobic respiration and photosynthesis. So in this problem, I'm going to go ahead and have you all pause the video and try to see if you can fill in the blanks. So there's quite a few blanks here, so make sure you leave yourself enough time. So hopefully you paused the video. We're going to go ahead and start with aerobic respiration and walk through that first. So in aerobic respiration, we have sugar that is oxidized to CO2, and that sugar of course is glucose. So glucose is broken down to CO2 over stages 1 through 3, and then oxygen, which is our final electron acceptor, is reduced to water. And this of course happens in stage 4 during the electron transport chain and chemiosmosis. Now on the flip side, in photosynthesis, water is going to be oxidized to oxygen, and so this happens during photolysis. And then we have carbon dioxide, or CO2, that is reduced to sugar. So again, this practice problem is showing us how these two processes are essentially backwards and inverse of one another. Our next practice problem is more of a prediction type, so an application question. So in this practice problem, I'm asking you all to predict what would happen if the Calving cycle is blocked. So essentially what I'm saying is, what if we can't do the Calving cycle? So maybe something is broken, maybe we have no RUBPs, one of the enzymes, perhaps Rubisco, has a mutation in it. And so what do you think will happen if the Calving cycle is blocked? All right, so let's walk through this together. So A states that NADPH levels will increase. So if we take a look at the end of stage 1 here, stage 1 produces NADPH and produces ATP. So if we stop this cycle is broken. Now we have stage 1 happening, and we eventually will end up with a ton of NADPH molecules, but none of those will be used. So if the Calving cycle is blocked, A is true, and B is the inverse of A, so B states that NADPH levels will decrease. We can cross that out because stage 1 is fine. Stage 1 will continue. And using that same logic here, because ATP is one of the products of stage 1 is continuing its process, and the Calving cycle is not working, so we're not using those ATPs. So those ATPs will increase. So again, E is the inverse of ATP, so we can cross that off. And then E and F talk about, what do you think will happen with the glucose concentration? Will it increase or will it decrease? Well, the part where we actually make glucose, where we make carbohydrates, is the Calving cycle. And that's what's broken. So if that is what's broken, there's no way that glucose can be increasing. It will be decreasing. So let me know in the comments if you follow my logic here and if you got the same predictions. So we're going to do another prediction type question. And in this case, the prediction question is going to be about the opposite. So what about the light reactions? What if these light reactions are broken and these light reactions do not happen? So same answer choices here. So A and B ask us about NADPH. So in the light reactions, we produce NADPH. That light energy is transferred to chemical energy. So if they're broken, there is no way that we can get an increase in NADPH levels. So what we will see is that NADPH levels will actually decrease. Same thing for ATP. Our other energy management molecule that we make is ATP, but we can't be making it right now because the light reactions are blocked. And so we're going to see that ATP levels decrease. Now for E and F, they're asking us about glucose. Eventually what will happen is the Calving cycle will use up all of the ATPs and all of the NADPHs, which means we won't be able to be making any glucose. So we will actually see that the glucose concentration will also decrease. So I just wanted to give you all a little bit of a flavor of the types of questions that I might ask in terms of application questions. So again, let me know if you have any questions on these or if you are confused. So the last two questions are ones that we have seen. So during day one of photosynthesis, I started our lecture with these two questions, and I wanted to see if your answers change. So let's go back to our potato question. So potatoes are full of starch, which is a polymer of glucose. And knowing what we know now about photosynthesis, where did the carbon come from to make this starch? It came from the air. And more specifically, right, it came from CO2. So again, let me know in the comments below if your answer to this changed at all, and if it did, how it changed. And then the last question we're going to talk about is our alternate lifestyles of cells. So we're going to go ahead and match these up as well. So for number one, an autotroph. An autotroph is going to be our plant cells. And so by taking CO2 and fixing it, they are, in a sense, self-feeders, and they're making their own glucose. For two, heterotroph. And that is going to be our animal cells. And then 3 is asking about producing O2 by photosynthesis. That's going to be our plant cells. And that O2 comes from stage 1, it comes from photolysis, and then producing CO2 by cellular respiration. So that is actually going to be both. And this is a really common question that many students often forget about. And not just students, adults too, actually forget that plant cells, not only do they have chloroplasts, but they also have mitochondria. So plant cells can do photosynthesis and can do cellular respiration. And specifically, this is referring to aerobic respiration. So again, let me know if you were able to answer this question both before and after. So we are going to stop here. This is the end of photosynthesis. So we are done with new information for our metabolism unit. And our next topic will be a modeling activity that we're going to do that's going to help us synthesize all of this information and just get more practice with it. And then after that, we're going to start our last unit, which is one of my favorite units because it's all about DNA and RNA and proteins. So I hope you all are excited, and thanks for hanging out with me. I hope you have a wonderful day, and let me know if you have any questions or comments or concerns. I miss you all, and I hope to see you soon. Bye, everybody.